A warm greeting to everybody here. So welcome to this YouTube channel called Gene Editors. So I'll be talking about the topic Arabinose Operon. So I'll explain you all what is Arabinose Operon and what is the mechanism that happens in the Arabinose Operon. I'm Nivetha from Bristol Jayanti College. So I'll be explaining you all what is Arabinose Operon. So we'll be seeing what is an operon and then the structure and the genes involved in Arabinose operon, then the mechanism, that is your positive regulation and negative regulation. Then we'll be having a summary on as to all of this. What is an Arabinose operon? So coming to the first thing, introduction. What is an operon? So your operon are the regions of DNA that contain a cluster of related genes, which are made up of your operator, promoter, and multiple genes present all together. So the operon theory is the concept of gene regulation, which was first proposed by Francis Jacob and Jacques Monod. These are the gentlemen who proposed this theory in the year 1961. Next is the Arabinose Operon. So before we get to know what is Arabinose Operon, let's see what is Arabinose. So arabinose is a 5-carbon sugar that can be used as an alternative carbon source for energy production in the bacteria E. coli. So this regulation of arabinose was studied by Ailes Engelsberg. This gentleman here, he was the one who studied the regulation of Arabinose operon in E. coli. So this Arabinose operon have various alternative names like L-arabinose operon, ARA operon, all of these. So Arabinose operon is basically a system that is carrying number of genes like regulatory, operator, inducer, and structural genes for the breakdown of your arabinose into xylulose 5-phosphate. So the structural genes are ARA B, ARA A, and ARA D. And these produce enzymes, metabolic enzymes like kinase, isomerase, and epimerase, respectively, which help in the conversion of this arabinose into your xylulose 5-phosphate, which then enters the pentose phosphate pathway. Next, coming to the structure of arabinose operon. So the structure of Arabinose operon is linear and it contains 4 specific genes along with the catabolic active sites. Here you all can see the regulator genes, the promoter genes, then you have the operator genes, then you have the structural genes, and then you have a catabolic active site. So this is your structure of your Arabinose operon. So the structural genes of Arabinose operon. There are 3 types of structural genes in Arabinose operon: the ARA B, ARA A, and ARA D. So their main function is to produce metabolic enzymes. So your ARA B produces kinase, and ARA A produces isomerase, and ARA D produces epimerase. So all these enzymes help us produce a polycistronic mRNA by converting by breaking down your non-glucose molecule, that is your arabinose. Next is the inducer gene. So your inducer gene, there is a site called as ARA-I where your inducer gene comes and binds. Okay, so in your arabinose operon. So the inducer molecule in our arabinose operon is arabinose itself. What it will do is arabinose will go and bind with the repressor protein and induce transcription to form the mRNA. So this Arabinose operon works positively when there is inducer present and works negatively when there is absence of inducer. Next is the catabolic active site. So in arabinose, there is an activator site termed as CAP. So what is CAP? CAP is catabolite activator protein, which activates the efficiency of transcription rate by promoting the effective binding of RNA polymerase to your promoter region. So when there is low availability of arabinose and high availability of glucose, ATP will not be converted into CAMP. So and then this complex will not come and bind to the catabolic site. So all of this regulations and all we'll study in the further slides. So you all should know there is something called as catabolic active site where this ATP cyclic AMP and your CAP protein together will come and bind there and bring about a regulation in your arabinose operon. Next is the operator gene. So there are 2 operator genes, that is your operator 1 site and operator 2 sites. Both play a crucial role in your regulation of your arabinose operon. Next, promoter region. Again, there are two promoter regions. One promoter is for your RAC gene, that is your regulatory gene, and one more promoter is for your structural gene, that is your BAD, that is your RAB, RA, and RAD. So your RAC repressor can exist in two types. That is your active P1 state. This state is found when there is absence of inducer, and there is inactive P2 state. This is found in the presence of inducer. So coming to the regulatory gene. So your RAC is the only regulator gene in the Arabinose operon. Okay, so this RAC, what it does is it encodes for a RAC protein. RAC gene encodes for a RAC protein. This RAC protein acts as a repressor. So what it will do is it will bind with your arabinose, and it can regulate your this RAC protein can regulate your Arabinose operon both positively and negatively. We'll see about it in the regulation. So coming to the regulation part. So regulation of Arabinose operon. So this Arabinose operon is not only under the control of your CAP and cyclic AMP activator, it also positively and negatively regulates through binding of your RAC protein. So RAC protein also has a role in the regulation of your Arabinose operon. So it can be regulated both positively and negatively, like our lac operon model. So before we get into how the regulation happens, you should know how the structure of RAC is. So your RAC protein is a homodimer. So what is homodimer? Homodimer is a protein that has similar proteins made up of, like it is made up of similar proteins, so same proteins, two units of same proteins. That is called homodimer. So this RAC is a homodimer. It has two units of the same protein held together. So this one monomer of this RAC protein will have two sites, two domains. One domain is for the DNA binding, and the other domain is for forming dimerization. So that is called as dimerization domain. You'll understand as to why these domains are important in the regulation part. Coming to the negative regulation. So here you all can see in the negative regulation, what happens is in the main role of your Arabinose operon involves the breakdown of your arabinose. So when there is no arabinose present in your system, in the bacteria system, then there is no need of switching on your Arabinose operon. So negative regulation should happen so that there is no formation of the products, your structural gene products. So basically what happens here is, in the absence of an inducer, that is in the absence of our arabinose, the repressor protein, that is your RAC, what it will do, it will go and it will be produced. Okay, the RAC will be produced, and this RAC repressor forms a dimer. Here you can see it forms a dimer between inducer 1, ARA inducer 1 and ARA operator 1, and it forms a dimer which forms a loop-like structure. So when this loop-like structure is formed, your RNA polymerase is not freed, like it doesn't have a free way to come and bind to your promoter region of your structural gene and form these structural genes. So hence here the transcription is blocked by the loop structure. This is how the negative regulation of your Arabinose operon happens. Next, let's see how the positive regulation of your Arabinose operon happens. So the positive regulation like the bad, that is your BAD mRNA, is formed by positive regulation. So the expression of ARA-BAD operon is activated in the absence of your glucose and presence of your arabinose. So your positive regulation can happen via two ways. One is by your RAC protein, and the other one is your cyclic AMP and your CAP complex formation. So let's see under each condition how it forms. So taking the first condition via RAC, how your positive regulation happens via RAC. So as I said, RAC can act as an activator also. So when there is Arabinose operon present, Arabinose sugar present in the bacterial system, what happens is it will go and bind with your RAC and it will form a complex. And then what it will do, it will come and bind to your inducer ARA1 and ARA2, and it will basically block this site so that the loop is not formed. Once the loop is not formed, then your promoter region is free for the RNA polymerase to come and bind, and then transcription can take place. This is how via RRC your transcription is happening, like the positive regulation happens. Next is via cyclic AMP and CAP complex. So as I said, this catabolic active site is where your catabolite activator protein with the forming a complex with cyclic AMP comes and binds. So when it comes and binds here, it when the glucose is absent. So when RRC, when is that RRC positive regulation happening is when there is a less amount of glucose and more amount of arabinose. That time, this positive regulation will happen via your arabinose RRC protein. This cyclic AMP thing happens when there is complete absence of glucose in the system, and there are high levels of your CAP protein and your cyclic AMP. And also your arabinose. So what happens is your cyclic AMP protein, cyclic AMP and your CAP protein will form a complex and it will bind to this site, this CAP site. So once it binds, what happens is due to this binding, the loop is broken. Okay, like in the negative regulation, when you saw the loop is there, that will be broken because the loop is formed between operator 2 and 1, right? So when this is blocking here, it cannot form a loop. So what will happen? Now again your RNA polymerase is free to come and bind to your promoter region of your structural gene, and it will bring about transcription. This is how the positive regulation happens in your Arabinose operon. Coming to the conclusion. So therefore, we can tell that Arabinose operon can both positively be regulated and negatively be regulated. So this is controlled by, this entire system is controlled by the activation and the repression. So the repression happens in the presence of a repressor protein, and the activation happens in the presence of an inducer. So we can conclude that the Arabinose operon is switched off in the presence of repressor and switched on in the presence of inducer. Thank you so much for listening. This is my references, and please do subscribe to this channel. Thank you.